Okay, so I'm going to close this one down. And so this was a paper that we were supposed to discuss earlier, but time got away from us. So like I said, I think we are going to be pretty good on track now. We're just one over. The chapter 13, we'll be good with doing it in 1.5 lectures, so we should be fine next week. We can catch up with that, and we're going to start it next week on Wednesday, and then the right before Thanksgiving. And then we can work on chapter 14 right after Thanksgiving. Okay, so let's go back into my module and I get to your chapter. Oh, not the chapter, the paper. So previously I've did a paper from my PhD time because it was a very simple paper to analyze and understand. That's why I had chosen that one. It was also not a very well-written paper. It was one of my first papers during my PhD. So, this one is a different type of paper. It has some positives and negatives. This is a new paper that we've just published with collaboration with Dr. Brian Nuckley and Corey Causey in the chemistry department recently. And I thought it would be a good thing for you guys to see how papers get published with new data, new findings, and also to understand different types of reactions and different types of techniques that are used in molecular and cellular biology as we try to find new drugs and try to look at their mechanism of actions. So I'm going to open this up. And I'm hoping that people were actually looking at it and not just, thinking about it. I don't know how many of you actually did. I want to open it like this. So it's just like big. Okay. Everyone can see the paper?
Yeah.
Perfect. Okay, so this one, Dr. Corey Causey and Dr. Brian Nuckley had been working with developing targeted cancer therapy using a specific peptide. So peptides is basically a tiny protein, not a big protein chain, a very small protein chain that could act as an inhibitor for the PRMT, protein arginine methyltransferases. And what they do is they inhibit specific types of methyl transfers, right? We talked about post-translational modifications, post-transcriptional modifications of DNA. That's what it's working on. It's working on those methylations to inhibit certain pathways that are activated in cancer cells. And so they found a couple of them and they had been working with them in vitro. They were not using actual cells. They were just using reactions in test tubes, and they saw that they seem to have activity that would be indicative of showing anti-cancer response. And so we collaborated with them to check it out in actual cells, cancer cells, to see if it actually did that work. So what we're going to do is we're going to go through figure by figure a little bit first. And I'm going to ask students to explain what they understand from that figure. And then I will bridge in the gaps. Okay. So the very first picture, the very first figure, it's a figure. Their title says protein arginine methyltransferase catalyze the transfer of methyl groups from S-adenosyl methionine to the arginine found in proteins. Okay, so what do you see visually looking at this figure? Again, this one is not a data figure as such. This is more giving the peptide structure and what it does, right? It's just a kind of proof of concept thing. So someone. Come on, guys. Don't all talk at the same time. I don't want to call on people, so I would like you guys to tell me yourself. What do you understand looking at this figure itself?
One thing that I noticed from that figure is that the type 1 versus the type 2 catalases either
Yes.
results in the asymmetric protein or the symmetric protein.
Exactly. So depending on whether it is a type 1 PRMT or a type 2 PRMT will lead to different conformation of that protein at the end of the day, right? Everyone good with that? That's the main figures, this figure's main message is that depending on what type of PRMT you're working with, it's going to give you a different product. And one of them may be more stimulated in cancer and one of them may be more in normal cells. And that's something that they talk about in their discussion and in their paper. So here they are with their results. The very first one is just, again, they had two different compounds, we call them, boring names, I'm sorry, P2 and P2A peptide. They were very slightly different in their conformation. If you look at their structure, you'll see they're very, very, very similar. They just had a little bit of a difference in their chains. But that little bit of difference made one of them a lot more stable and a lot more effective than the other. And so our very first figure down here, this is in vitro. This is saying compound P2 inhibits PRMT1 at low micromolar concentration and exhibits selectivity over PRMTs. So what is it telling me? Let's go through this figure and tell me, what do you understand from these graphs? What do you see when you look at this particular figure? How do you read these graphs? So for the top two are going to be, I think, your PRMT2A peptide, and the bottom ones are PRMT2, just the P2 and P2A. A and B is P2, P2A, and C and D are P2. What do you find? Is one more effective than the other? Number one, which one is more effective than the other?
P2 is more effective.
Yes,
P2.
and why is that?
It exhibits more it's more inhibited than
It's more inhibited
P2A.
at a lower concentration. Good. So if you remember our cell viability module, we talked about a specific value that we can calculate. What would that be? What do we call it if we are looking at efficacy of the compound? What is one of the things that we can calculate to see which one is more effective? We talked about calculating that value for our compounds as well.
Confluency?
Say it louder.
Confluency? No.
No, confluency is measure of the cells. There are no cells in here right now, right? This is just a reaction in a tube. But there was a thing called IC50, if you remember. What is that when we talk about IC50? So IC50 is typically when the reaction or the cells are dying at 50%, like it's inhibiting it by 50%. So if we do that for the very first, for P2A, that value comes out to be about 62.5 micromolar. But if we do that for our P2, that value is only at 7.8, right? That's a big difference. What's the second part telling us? So it tells us that this is a very specific inhibitor for PR. No, it doesn't tell us that. But if we just look at A versus C, all it tells us is that for PRMT1 inhibition, P2 is a lot more effective, right? It's about 10 times more effective than P2A. What does B and what's the purpose of panel B and D? And by the way, it did say down here too, IC50 plots of compound P2A. I think we could have just read that too. But besides the point, what is the purpose of panel B versus D? Why do we need those? It shows that PRMT is inhibited. PRMT1 is inhibited. Why look at PRMT5? What does that tell you?
that it also works on different structures of the same
it?
compound?
Does it work on the same, on different structures or different PRMTs? Is it as effective? It doesn't work at all.
Yeah, I was going to say, showing that PRNT5 shows no inhibition, so it shows that the difference is
So
specific
yeah,
to
so it
the-
shows that P2A is very specific to PRMT1. It's not doing anything to PRMT5. What about P2? Is P2 also is specific the same way?
No.
It's not bad, but it's not as specific for sure, right? You're still seeing like 40% inhibition. You have to go up to 500 micromolar, so you have to go really, really high concentration. But definitely it has a little bit of leakiness, right? It's maybe not as clear. So now we get to actual cells. You guys should like that one. So this one, you guys can see it clearly here. I know it's a little bit small, but I'm trying to make it as large as I can without like affecting it too much. So this is the one of the figures that we provided to them. There are 3 different cell lines in this one. There is MDA468. What type of cell line is that? You guys should know.
Breast cancer?
Breast cancer cells. HCT116 plus P53. What kind of cell line is that? HCT116s are colon cancer. Good job. HEPA-RG. What type of cells
Liver
are those?
cells. Liver
They
cells.
weren't, they're liver cells, they are non-tumorigenic, remember? They do not cause tumors the same way. They are not cancerous. So that's the difference between them. Looking at panel A, what conclusion can you draw? So panel A is the pictures, cell viability, looking at untreated cells, and then looking at 3 different concentrations. And panel B is the quantification of that, okay? So looking at that, what do you find? What conclusion can you draw from this figure? This is an example of the type of questions you will have on your quiz coming up for the lab as well. So looking at this figure, what conclusion can you draw?
HCT and MDA cells were inhibited much more than the HEPA.
Yes, exactly. That the normal cells were not affected, but the cancer cells were. And that tells you what? Is this a good targeted therapy? Yes, it would be a good choice as a targeted therapy. It's not the best, but it's good in combination with other things. So here is the next one. This is looking at cell viability over time. Okay, so this is looking at cell viability over time. Again, you have your cancer cells up here, and then HMECs are human mammary epithelial cells, just normal mammary epithelial cells. HEPA-RG are again those liver cells. And we are looking again over here at relative growth over time, right? So let's look at one of these, MDA468, and look at the untreated, the blue line, right? That's your typical growth. What do you see when you start adding the P2 compound?
It inhibits the growth as you go up in dosage.
Yes, so it is a dose-dependent response. Do you see the same thing for the second cell line?
Yes.
Yes, even more dramatic actually, right? And then let's look at the normal cells. What do you see with the normal cell lines?
It doesn't affect them.
It does not affect them. Good job. So this would show again that it is specific over time and over dosage to cancer cells and does not affect normal cells. And again, looking at dead cells using trypan blue or another method like that, you can again see the induction of dead cells in here, right? More dead cells in a dose response manner. And this is only showing for MDA468, but you could do that for all of them. Okay, last image before we go, because I think you guys should be getting ready to go. This is another assay that we do. So, it's easy to grow cells in culture in a single plate, in a single layer, and look at them. But cells don't grow in a single layer inside our body. They grow as balls and tumors and organs. So we have to mimic that somehow. So there's a special technique called soft agar colony formation assay. What we do in there is we embed cells in this agar kind of matrix, like jello-like matrix, and they grow balls. So they grow like little tumoroids, right? They're tumor balls. And so in a normal cancer cell line, right, if you just take a cancer cell line and you make these, you would get these colonies. You see these little balls? These are not one cell, these are a bunch of cells, 100, 2,500 or more cells in each one of these balls, and they will be bigger and more robust in some cases. And then if you add your compounds in it, you would, if it's going to inhibit tumor formation, you would see less and less colonies. So here we have that data for two different cell lines, again, our two cancer cells. What do you find? What
It
does it show?
inhibits the growth.
Yes, that it not only kills the cells in a monolayer culture, but also inhibit the formation of tumors in the colony formation assay. Does that make sense? Higher doses lead to decreased colony number. Yes. Okay, one more actually. I forgot about this figure. This is a good one for you guys to kind of look at as well. This one now is trying to understand what caused it. How is it doing it? This is mechanism of action. So there are two different, remember, if we remember from our looking at the different ways cells die, we talked about necrosis, autophagy, and all that good stuff, right? So this is looking for markers of those. So let's just focus over here on these morphology images first. And look at, these are again looking at some cells, and we are just showing you one of the cell line in here, over here, right? You have untreated cells, and then you have cells, these are all MDA 468 at 48 hours, and we are looking at morphological markers in there. What are some things you notice? What are the red arrows pointing to? Look at the red arrows. What are they? Blebbing.
Explosion.
Yes, so
Blebbing.
blebbing is a marker for what type of cell death? Apoptosis, good job. Now look at the black arrows. What are they pointing to?
Necrosis.
They're vacuoles, but they're like lots and lots of vacuoles. So they're not necessarily necrosis in those cases. The cells may not be fully necrotic. What is another way that you get lots of vacuoles in a cell? Autophagy, good. And so that's something that can happen as well. So the most common reasons looked like were either autophagy and apoptosis. For apoptosis, we already know, you guys have worked on that. Caspase-3 is a very good marker to examine. And so let's look at these markers. So this is looking at caspase-3 activity at 24 and 48 hours of treatment. What do you find? Again, you have three cell lines. You have MDA, HCT, and HEPA-RG, the liver cells. What do you find for caspase-3 activity? So you're looking at panel B and C. It increased with dose in MDA. Did it increase in HCT as well? A little later maybe, but it has a really high activity in 48 hours, no? Yeah, so both MDA and 468 show robust increase in caspase-3, which would indicate that they are dying from apoptosis. Good job. Now, panel D, remember one other morphological marker of autophagy is reduced cell size. You guys remember that they become smaller and smaller over time, right? So in panel D, we looked at images and looked at cell sizes and just took averages of all the cells that were there. What do you find for HCT and 116 for the drugs? What are they doing? Are they showing some reduction in cell size? So this is looking at relative cell area. Yeah, they're shrinking, and that's not present in HepARG. So that will also indicate that there is autophagy happening. So to prove that, we looked at a molecular protein, a protein marker, LC3, that is a classic marker for autophagy. And so if you see it higher in the levels, that indicates that there is autophagy happening. And so if you look at this, it is not the most dramatic increase, except in MDA, you see a lot of it. So it's probably showing that MDA, the main focus might actually be autophagy. In HEPA-RG, there's very little, but HCT3 also shows an upregulation, but not to the same extent. So in HEPA-RG, you wouldn't see too much of a response. MDA seems to be more focused on autophagy, and in HCT116, the death seems to be more focused on caspase. So it does have some baseline, you're right, and that's a very good question. These are cancer cells, and so there may be automatically happening, some background autophagy happening because of their high growth rate already present there. But there's definitely an additional upregulation compared to that. Good job, very good question. But down here is a loading control to just show that we loaded about the same amount of protein in all of them. Good. Good discussion. I am very happy with some of the responses. So yeah, hopefully that helps you see how we, build our research story and come up with our ideas. As you do the work in also the lab section and do those posters, that's what you should be thinking about. How do you make that story? How do you go from, like, this is our problem, this is what we think, this is what we found, this is our answer. Okay? Okay, guys, good job. I'll see you guys in lab or next section. Take care.